Hi, APB4 folks. In this video, we're going to cover the intricacies of S phase. Specifically, how do we copy chromosomes into identical copies of themselves? So, as you can see in the upper diagram, we're going to start with a really long eukaryotic chromosome, maybe 100 million base pairs in length, and we're going to start peeling the two strands apart. And in the end, after those two strands are pulled apart, there will be a new complementary strand on each one of them, resulting in two copies of the original chromosome. Now, because each chromosome has one original copy and one new complementary strand, this is called semi-conservative replication. Now, this might seem obvious to you that you peel the two strands apart and then put a new complement on them, but this wasn't entirely clear literally 70 years ago when this was first being studied, which is why you're required to memorize this term, semi-conservative replication. I don't want to spend too much more time on it, but do know the two strands are separated and then a new complement is added to each of the strands to create our two new chromosomes. Okay, so our steps. The first step is to peel the two original strands apart. I'm going to call this denaturing because it's a term you know from understanding protein structure. And the enzyme that's responsible for doing this is basically a motor called helicase that's going to start motoring away from itself. Two copies are going to go in opposite directions, breaking the hydrogen bonds between the base pairs, allowing you to form this thing called a bubble. So this is going to be a replication bubble, which you can see between the two green blobs. There is an additional set of proteins called topoisomerases that have to deal with supercoiling downstream. I'm not going to cover them in this video. Feel free to look that up on your own time. Okay, so let's imagine that these helicases keep motoring along in opposite directions, breaking the hydrogen bonds. Quick reminder, these two strands are anti-parallel, running in opposite directions, 3 to 5, 5 to 3. This leads to our next problem. So you might imagine that there should only be two enzymes required to do this whole process. You'd imagine one to peel apart the strands, that's our helicase, and then the other enzyme you might imagine being necessary is something that makes a new strand of DNA. And so we could call that enzyme DNA polymerase. There is a DNA polymerase. There are actually dozens of types of DNA polymerases out there, but it has an incredibly serious limitation to it. The limitation is DNA polymerases can only add to the 3' end, that's the sugar end, the deoxyribose end, of an existing strand of DNA. All right, so again, DNA polymerases can only add on to the 3' end of an existing strand of DNA or RNA, actually. So I'm going to draw in what actually happens, which is weird and confusing, and the step is called priming. So on each of the two strands, you're going to have to add a landing pad for our polymerase, and this landing pad is actually made of RNA. The enzyme that makes it is RNA primase, or just primase for short, and it puts down 20, 30 base pairs or bases of RNA here. So the nucleotides are added in a 5 to 3 direction as always. And what makes RNA primase special is that it can start a new strand from scratch, and it does. Once you've got this little landing pad, this RNA primer, on the 3' end of that primer, the DNA polymerase can then bind and extend using DNA nucleotides. And so we're just going to chug along at the end of that primer and extend that strand all the way until we're running into or running right behind the helicase. All right. So again, the limitation of DNA polymerases is they may not start a new strand. They must extend an existing strand, which can be made of RNA or DNA. In this case, we're going to start the strand with RNA and then extend with DNA nucleotides. If you're at this point, you might think we're done because it looks like we're done. It looks like the polymerases are just going to keep chugging along behind the helicases and we're all set. But if we fast forward in time and we look at what the replication bubble will look like a few moments later, you will see that there is a problem. This is the same strand that we were just looking at just a few moments later. the helicases have continued motoring in opposite directions, exposing more single-strand DNA. Okay, they've peeled those two strands apart a little bit further. And the polymerase at the end of the DNA strand now is going to keep on following that helicase. But hopefully you'll see that there are two areas behind the primers that have not been replicated and will not be replicated unless we do something special. So this is where things get really annoying. We're going to have to reprime on a regular basis on one of the two strands at each of these sort of places where the helicase is acting. We're going to add new primers using RNA primase, and then the polymerase is going to have to jump on again and extend that primer away from the direction the helicase is running. So again, one polymerase is going to be right behind the helicase, and that's the one we're going to call the leading strand because it's sort of going fast, or it's ahead of the other one. It's right behind the helicase. Now, on the other strand, the polymerase is moving away from the helicase, and it's always falling behind because the helicase is always exposing more single-stranded DNA as the polymerase is moving away. Now, you might think that the polymerases are running in opposite directions, and technically they are running in opposite directions, but keep in mind they are still just adding to a 3' end of the strand. They are still adding onto the 3' end of a DNA strand, because that's the only thing they can do. It just happens to be that the strands are anti-parallel, which is why the new strands are also being extended in opposite directions, or so it seems. All right, so looks like we're done, except we're not. So here's the leading and the lagging strands, and here's what it's going to look like a few moments later. Okay, so imagine that we're not even going to continue with the helicases. We're just looking at the currently replicated stuff. We still got a couple problems here. One is that the new complementary DNA strands aren't all DNA. They've still got some RNA in them. Those primers are in your DNA. And if you see the little gaps, that's where the DNA polymerase was chugging along, and then it ended up running into the RNA primer. The DNA polymerase only has the ability to add new nucleotides. It can't put backbones together from existing fragments. So we're going to need two new steps here to finish this off. One, we're going to have to replace those primers, and we're going to use another DNA polymerase. You don't need to know the details of this. It just happens to be a different version of a DNA polymerase, and it's going to remove the RNA nucleotides one at a time and replace them with DNA nucleotides. Uproot the RNA, replace with DNA. Uproot the RNA nucleotide, replace with DNA, one at a time. Once we've done this, we then have to just quickly staple the backbones together of these fragments, which you may have read about are called Okazaki fragments. So we're going to now take our enzyme DNA ligase, and that's going to take those fragments and just finish the deal. All right, put the fragments together in terms of their backbones. Just connect the exposed 3' and 5' ends there. Okay, in the end, we have separated the two original strands that were anti-parallel, built two new complementary strands on them, and now we have two identical copies of the same chromosome. They are called sister chromatids. That is a strange and annoying vocab word that you are required to know, and the key thing to know is they are identical. Unless there has been some polymerase error, which does happen, and that's called a mutation, these are identical copies. These are not like your mom or dad's version of the same chromosome. These are identical. And the other thing you should know about it in terms of the cell's division cycle is they stay attached. These are actually connected by proteins, and they do not freely float away from each other. They are attached to one another until you complete the checkpoint in the middle of mitosis and then pull these chromatids apart. That's the sort of defining moment of mitosis that you all have seen before. All right, now we're at the end of our discussion of S phase. In a future video, we'll talk about how we can actually replicate this process in short bursts in a test tube. All right, hope that helped everybody. Bye.